Hello, this is Nick Cap, and again, we are doing the 20-minute wrap-up of a lecture that was we first seen this past Thursday, and we are looking at basically the origin stories, and the origin stories of all of us, the origin story of life. Let me set this up here. Let's go back previously. And so again, we're looking at the origin stories of life. This is a cell biology class. We're mainly going to be looking at cells. With that, what we wanted to say is life comes from only other life. And so in the cell theory, cells only come from cells. And what we got to say is when life first began, there had to be that one or two chances where life came from non-living things. And it kind of blows me away in that when we've tried to do this in our laboratories, we can't do this. And so how do we look at this? And again, there's a number of different things that affect this. Geological events can change the course of evolution. We are changing the course of evolution around now. Again, life that's on this planet has changed this planet from what it used to be before. And so basically what it is, the constant that we have on this planet is we are changing. Nothing's going to stay the same. Sorry, it's a biology class, so I have to show you pictures of dinosaurs and bones and pictures of possibly what it was like before. Again, these are just stromatolites that are in here, and this is an indication of life already kind of going on in this area. But what I wanted to do is to kind of give you a little concept of before we look at living things, life only comes from life. What had to have happened, or what were the possibilities? What are the breakdowns? Again, I'm a reductionist. How do we look at these things? And so what had to have happened is we had to have some way of an abiotic synthesis of our organic molecules. Today, amino acids only come from living things. We don't see wholesale production of amino acids out there in the environment. If it's happening at such small values that we can't read it. Again, we needed to have the joining of those molecules into macromolecules. Those macromolecules need to be packaged in unique units where these reactions can occur, and then we need an origin of self-replicating molecules. Again, if we can self-replicate these macromolecules rather than have them randomly be produced, then that would make this system much more efficient, and then it would also allow for, I can't write it down here, evolution, which again would make things better. Again, the Earth was very different about 4.6 billion years ago. Is the age of water. The Earth's early atmosphere consisted of water vapor, chemicals released by volcanic eruptions, things like nitrogen, nitrogen oxides, carbon dioxide, methane, ammonia, hydrogen sulfide. What we cannot see there is oxygen. And so it's really kind of great. This is a graduate student at the University of Chicago put all these materials and put a spark across this, and you would say, hey, methane, ammonia, hydrogen, that's a recipe for a big boom. But without oxygen, it won't oxidize. You won't have that big boom. When we did this, we got simple molecules. And again, this is a picture from your book showing you a little bit about this. These are, again, when we look at all this materials that are in here, those molecules can go up into polymers by actually dripping them onto some clay-like materials. And so here's the precursor just sitting there, and this is time in minutes, and then we can get some polymerization of it, but it happens much faster in the presence of something like clay. So that's basically dirt. So again, the idea that we had an early reducing atmosphere, lots of hydrogen in the atmosphere, may not be necessarily right. There's some evidence that proves that. But again, we have these things here called these smokers, dark smokers on the base, and then again, we have reducing areas inside of here. And so even though this area here may not be reducing, that is over there, and we have much change. Again, when we look for amino acids, everywhere we've looked for amino acids, we've seen them. Amino acids, again, from rocks, meteorites from outer space, have amino acids in them. Again, there's a process that we call panspermia, where some people think that life came from outer space. But again, this had to happen somewhere. Probionts are aggregates of abiotically produced molecules surrounded by a membrane-like structure, which looks a lot like a cell, and I'll kind of show you that. So this is a probiont. And so again, we take phospholipids, put them in a jar, and mix them up. They will produce this structure, and I talked a little bit about how that happens in class. Again, if we trap certain of these amino acid polymers inside of there, they can act as enzymes, and we can actually get activities. And people have done these kind of experiments that we have on there. Again, the last thing that we need is self-replication. Not of the entire probiont, but of the macromolecules that are in there. And what we see, a unique molecule of RNA, and even today, RNA can act like an enzyme, RNA can act like as an enzyme and act what we call ribozymes, and RNA can facilitate its reproduction. So it's really kind of great, neat stuff. So what we had is life that was able to reproduce. What I'm going to do is I'm skipping over to this slide right here, is I do want you to know the timeline, and we can use it a little bit like a clock. Again, the Earth is roughly about 4.5 billion years ago. Roughly about 3.75, we start to see the what we would call living things, the prokaryotes, and there's a number of reasons why we think this time. At roughly about 2.75 billion years ago, we see the production of oxygen in the atmosphere. Oxygen allows us to do oxidative respiration, in which case we can get a lot more energy out of the cells. Single-celled eukaryotes, roughly about 2 billion years ago. Multicellular eukaryotes, about 1.5 billion years ago. And basically animals and all the organisms that are alive, roughly about 500 million years ago. So we've been around for a long time. The one thing I do want you to know is that we have this thing called the oxygen revolution. Oxygen is, all the oxygen in our atmosphere is of biological origin. It came from basically water, and we'll talk about that. And so roughly about 2.7 billion years ago, oxygen began accumulating in the atmosphere and causing rust on iron-rich terrestrial rocks that we could see. These are the stromatolites that we have. This is actually photosynthetic, and so we don't think about these as being plants or being like plants and producing oxygen, but they do. And what we see here is that oxygen actually caused the precipitation of iron out of the fluid, and so some of our more rich iron ores are actually due to this structure. Another thing that we had, again, so what we're talking about is refinement. For some reason, we had life come, and we had these early simple prokaryotes, and evolution can work on those simple early prokaryotes. Again, oxygen was a great thing. It was a bad thing for a lot of cells. It was a great thing for a lot of cells, in that those cells were able to use oxygen in a process that we call oxidative respiration in order to make more energy. Now, what we see here is a combination. And so a lot of you are thinking of biology is that we had that first protogene, or we've had that first organism, and we are all descendants of that first organism. We had that change. But we're actually, what we may see is that we had several organisms both develop, and for some reason these organisms kind of came together. And so some people, when they talk about the tree of life, they talk about the bottom of the tree of life kind of being a little bit of a circle, and that we didn't have linear transfer of genes. We actually had material swapping out, and actually endosymbiosis is that we probably had whole organisms swapping out. And so here we have a simple prokaryotic organism. As that organism got big and it was able to produce proteins and stuff like that, that organism found that it was actually much simpler to engulf, rather than engulf and eat and just use the materials of a heterotrophic prokaryote, that is one that's able to undergo oxidative respiration. If it took it up and enslaved it and trapped it, it would be much better. And actually for photosynthetic prokaryotes, we see that as well. And so the plants that we have, actually there was a couple of endosymbiotic relationships. And what we see, these are symbionts, and we'll talk about it in a second, and that some of these genes are transferred over to the nucleus, and some of them weren't. These organisms, these organelles, are not able to live on their own, But again, they still have some of the characteristics, and so these characteristics are, we have similarities in the inner membrane functions to prokaryotes. Division is similar, how they divide, how these organelles transcribe and translate their own DNA. Again, mitochondria and chloroplasts have their own structures for doing that as well. Again, the Cambrian explosion, which is roughly about 500 million years ago, 500 million years ago, basically all organisms that are alive today, that basic pattern is there. And I won't go into that in as much detail in here. Again, here is kind of what we think is a fossil of a early developmental process. Again, what we need to stress is that evolution is kind of like tinker. It's a process in which whole new forms arise by slight modifications of the existing forms. And so here's the timeline that I gave you on the events. Kind of know what these events are and be able to describe them. How do we get such big changes? And we'll talk about DNA and we'll talk about what genes are, but we're going to talk about these things called homeotic genes, which determine the basic features as like wings and legs and how they develop, and we'll explain what these are in a little bit detail there. But now, for now, you just kind of need to know what they are. And kind of what's happening, we think is what's happening is that these genes are duplicating, and then they go on and then they change a little bit. Oh, I got somebody waking up there. And so we'll talk about that a little bit more. But again, it's the regulatory genes, the genes that control how an organ grows and develops, that can make very big, slight little changes can make very big changes. And so again, going back, we can go from an organism that has many legs, where each segment has a leg, to an organism that has relatively few legs, by changing these regulatory genes. And we will hear more about that a little bit later. Again, things that we need to discuss is how humans continually alter our environment. Is that a bad thing? Is that a good thing? Or is that just a thing that happens and that we need to be prepared for? And so, again, I hope you enjoyed this origin story, and we'll see you next time.